Okay, so welcome to this next video in which we are discussing the adaptive immune response. Okay, so we now want to talk about the cell-mediated response. Okay, so we are now discussing a intracellular pathogen, such as a virus, which is within the cell. It's infecting cells rather than just living in the tissue fluid around cells. Okay, so we've discussed that this will lead to antigen-presenting cells presenting antigen fragments from these intracellular pathogens on both MHC class 1 and MHC class 2. Because MHC class 1, because the intracellular pathogen will infect the professional antigen-presenting cells, and then the antigen will be chopped up and presented on MHC class 1, and then MHC class 2, because the intracellular pathogen, even though it's intracellular, when it spreads between cells, it has to temporarily go extracellular. And in that phase, it's vulnerable, basically. It can be phagocytosed by these professional antigen-presenting cells. And then, once it's been phagocytosed, its antigens will be chopped up and then presented on MHC class 2. So phagocytosis involves, ends up with antigens being presented on MHC class 2. Just having a pathogen intracellularly leads to the antigen fragments being presented on MHC class 1. This leads to the activation of CD8 positive cells by the antigen fragments which have been presented on MHC class 1, and also CD4 positive T cells. Okay, so we're going to start with the CD4 positive T cells, and then we'll go finally and look at these CD8 positive T cells. So remember, once you got activation of the CD4 positive naive T cell, it then differentiated into a T helper 0 cell, and then divided to form a whole population of T helper 0 cells within the cortex or the paracortex of your lymph node. Okay, now if there was if the pathogen that we were launching this against was an extracellular pathogen, the T helper 0 cells then differentiated into T helper 2 cells. However, we're now discussing intracellular pathogens, so we're now going to assume that they have differentiated into T helper 1 cells instead. So let's discuss what T helper 1 cells do. Okay, so this is now the cell-mediated response. So the CD4 positive T cells will differentiate into T helper 1 cells, okay? And these T helper 1 cells start producing a certain molecule which instructs macrophages, basically, to become brutal, okay? So they start producing a molecule which is very, very nasty molecule, known as interferon gamma, and this is really the command to get brutal. It's kind of before the macrophages were kind of being gentle, whereas once you start producing this is basically the command of I want no prisoners, don't take any prisoners, just deal with it, basically. It's a brutal command that gives these macrophages new powers. It gives them the right to do horrible things. Okay, so it's going to go out into the bloodstream, so you're going to get loads of these T helper 1 cells that are producing a huge amount of this interferon gamma. And by the way, interferon gamma is often abbreviated to IFN gamma for short. And this is going to go into the blood, and it's going to go to the macrophages in this site of infection. So remember, the acute inflammatory response had led to the recruitment of a huge number of monocytes from the blood, and they've now differentiated when they went into the interstitial fluid into macrophages. And it's going to go to these macrophages and tell them just to kill like mad, basically. It gives them new powers and they can be brutal. Okay, so it activates the macrophages. So what examples can I specifically come up with for this activation of macrophages? Okay, well, there are certain intracellular pathogens that are bacteria which live inside macrophages. Okay, so let me show this. So if we've got a nice big macrophage here, okay, then there are certain bacteria which can live within the phagosome, basically. So the macrophage engulfs the bacteria, thinking that it's going to destroy it, but then the bacteria tricks the macrophage, goes to all sorts of length to stop the lysosomes from being able to fuse with the phagosome. Okay, so this here is the phagosome with the bacterial cell within it. Okay, so I'll colour in the bacterial cell in red here. Okay, and basically certain bacterial cells can then stop the phagolysosomal fusion, as it's called, which is this fusion between the lysosomes, which contain these lysozymes, which are going to digest the bacterium if they do get into the phagosome. Okay, and it's basically going to release a huge number of molecules to stop this from happening. Okay, so what sort of bacteria can do this? Okay, well, the most famous examples are Mycobacteria. Okay, and within the category of Mycobacteria, the main example that is capable of doing this, or the main example that's most famous, is Mycobacterium tuberculosis. And let me just move this up a little bit. So Mycobacterium tuberculosis, which is the causative agent of TB. Okay, so Mycobacterium tuberculosis, one of the most dangerous and deadly bacterial infections within the world. It claims that, I think it is the most deadly bacterial infection in the world. I think there's over 1 billion people infected with tuberculosis worldwide, so it's extremely dangerous and it usually does kill you in the end. So it's not a nice bacteria as well, and it's extremely difficult to treat with antibiotics as well. Okay, so Mycobacterium tuberculosis, it goes inside your macrophages, stops the macrophages from being able to kill it, and then just lives inside them for ages. Okay, now another example, another type of bacteria, well, another collection of bacterial species which are capable of doing this are Listeria species. And a specific Listeria species that is most famous of the Listeria species is Listeria monocytogenes. Okay, which causes listeriosis. Okay, Listeria monocytogenes, rather. Okay, and this is a I really struggle with this word. A gastroenterological bacterium, so it will cause gastroenteritis, basically. Okay, so both of these species are capable of going into macrophages and then stopping phagolysosomal fusion, and because that's such a good word, I will put it in. Phagolysosomal fusion is the fusion between the lysosomes and the phagosome membrane, okay, which leads to the lysozymes being dumped into the phagosome. Okay, right, so when you activate these macrophages with interferon gamma, basically it's going to tell the macrophages to get really brutal, and they can then start, at least in principle, or at least to some degree, starting to clear these infections. I think it's more effective in the case of Listeria monocytogenes than it is in the case of Mycobacterium tuberculosis, but the interferon gamma helps the macrophages, and we are looking into whether interferon gamma would be a good treatment potentially for some of these infections, particularly things like Mycobacterium tuberculosis. Okay, right, so interferon gamma sends this signal out to macrophages to get brutal on intracellular pathogens, basically. Okay, in addition, the other portion of the cell-mediated immune response involves these cytotoxic T cells, which are also to some extent activated by interferon gamma from the T helper 1 cells. So let's go back to our discussion of CD8 positive T cells. So we had got our CD8 positive T cell activated by an antigen presenting cell, which was presenting this antigen fragment on the MHC class 1 protein complex. Okay, right, so what is this this CD8 positive T cell going to do next? Okay, so let's have a look at this. So basically, it's going to undergo an activation process, okay? So it's already undergone the activation process. So the interleukin-2 has bound to the interleukin-2 receptor, okay? And shall we draw this? I'll try and fit it in on the bottom here. So here is our initially CD8 positive T cell, okay? Our CD8 positive naive T cell, okay? And now what has happened is that it's finally received this signal free in the form of interleukin-2. And what's going to happen is that it is going to differentiate into a type of cell known as a cytotoxic T cell. Okay, and this is often abbreviated to T for T cell, and then you put a C underneath to denote cytotoxic. So it's going to differentiate into a cytotoxic T cell. Okay, so remember, we got the activation of the CD8 positive naive T cell in the lymph node, basically. So it was within the cortex or the paracortex of the lymph node. So far it has not divided, it's just differentiated into a cytotoxic T cell, and now what it's going to do is it's going to divide. So now division is going to follow, and you're going to make a whole clonal population of these cytotoxic T cells. So here's one, and again I'll draw 3, but of course you'll end up with millions of these things. Now, what are they going to do? Well, their primary role in the immune system is to kill cells that have been virally infected. Okay, I'm just going to label this process as differentiation. Okay, so they are going to be now released into the bloodstream, and they are going to go and find cells which have been virally infected and kill them. So more generally, they're going to go and find cells which have an intracellular pathogen within them, and they're going to lead to the apoptosis of those cells. Okay, so let's get another piece of paper and continue this discussion. Okay, right, so if you are a cell, if you are an ordinary cell, basically, you are still capable of antigen presentation, and finally I'm going to explain to you how all cells are capable of antigen presentation. So, if you have some sort of intracellular pathogen within you, which the main example would be viruses, but there are a huge number of parasites which are intracellular pathogens which live within cells. For instance, malaria lives within cells, okay, Plasmodium species. So, viruses is the main example we'll use, but as I say, there are other intracellular pathogens. Then what's going to happen, basically, is you're going to have all sorts of antigens of this intracellular pathogen within you. So I'm just going to give you a nucleus because you look kind of disturbing without it. Okay, so you get these antigens from the intracellular pathogen, and I'll just denote this in blue here. And basically, all cells in the body, well, all nucleated cells, and I should add this, all nucleated cells are capable of antigen presentation. So what they will do is they will take the antigens of the intracellular pathogen, they will break them down, and they'll get fragments of these pathogens, and they will mount them, they'll display them, they'll present them on major histocompatibility complex class 1. Okay? Now, the difference between these normal cells and the professional antigen-presenting cells is that these cells do not put co-stimulatory molecules on. Okay, so they just present the antigen fragment on the surface with MHC class 1. Okay, so here is the antigen fragment, like so. And now, what will happen is that the cytotoxic T cell will come along to this cell, which has got this antigen fragment on its MHC class 1, and of course this cytotoxic T cell here recognizes that fragment. it has got a receptor which binds exactly to that fragment, and it also has a CD8 protein which will bind to MHC1. Okay, so this is CD8 here, and this is the T cell receptor here. And basically, when this happens, what will happen is that the T cell will say, right, this cell has been infected with this intracellular pathogen that I am supposed to be eradicating, and what it will say is, we need this cell to commit suicide. It's the only way, basically. This cell needs to sacrifice itself to save the meta-organism, basically. It needs to sacrifice itself to save 100 trillion other cells that are genetically identical to itself. Okay, so by saving itself, it saves itself. Well, sorry, by sacrificing itself, it saves itself, basically. Okay, right. So that is what cytotoxic T cells are going to do. They're going to trigger apoptosis in these cells which have got the intracellular pathogen within them. So this is going to trigger apoptosis. Okay, right. And this apoptosis is induced via the extrinsic apoptosis pathway, okay, so via the FAS ligand system, which I have a whole video on in my cancer playlist if anyone's interested. Okay, the final thing to say is that CD8 positive T cells will also create memory T cell, cytotoxic T cells will also create memory cytotoxic T cells. Okay, so not all of them will actually go around killing the cells. Instead, some of them will remain in the blood as memory CD8 positive T cells. Okay, and then if they get, if the antigen is presented to them again, they won't require the co-stimulatory molecules to activate them again, and then they'll divide and produce a huge amount of cytotoxic T cells. Okay, so that is the cell-mediated pathway for the adaptive immune system, which is aimed at killing intracellular pathogens.